labor in bee colonies, which shows you that there was more than just an individual that was important to the survival of the species. He also was the first to show that social information really induced massive gene expression changes in the brain of bees. But I know Gene particularly well because Gene's at the University of Illinois where I am, and Gene is not only an incredible or maybe credible scientist, researcher, but he is an incredibly, I guess I have to quit using that word, generous advisor and a member of the community at the University of Illinois that makes the university what it is today. So with that, I want to welcome you, Gene, for your talk.
Well, thank you very much for that kind introduction. And thank you, Larry, for the invitation. It's really a pleasure to be here. Larry has been a real strong supporter of the institute that I direct, the Carl Woese Institute, a valuable advisor and supporter, and we appreciate all of that. I'm really delighted to be here today to talk about honeybees, and Bryce really set things up for me very nicely in that video when he spoke about the fact that part of this amazing project is to build on the angels inside of all of us and get us to do something bigger than what we can do as individuals. Well, I'm here to tell you that honeybees and other social insects have been doing just that for 150 million years. And the essence of the problem is that honeybees have a brain the size of a grass seed, and so relatively limited capabilities as individuals, but together they have been able to create a society that's been around for a long time, that's been very successful, and we want to understand that. So that is an important aspect of biology in general, but in the context of this symposium where we're looking at health, the question is, so what? What kind of relevance does this sort of thing, do these findings of societies that are formed by insects, what kind of relevance might there be for human health? That's the subject of today's talk. Let me begin with Carl Woese. Many stories in biology begin with Carl Woese, and here is a slide that shows the history of life on the planet. Carl made signal contributions down in here in envisioning with amazing prescience a pre-Darwinian life, if you will, characterized by transfer of genes horizontally rather than descending them, so your children getting them is our typical vertical inheritance. Here we talk about lateral transfer of gene. So in this early stage of the history of life, these are billions of years over here, we switch to millions here, life could be characterized as a kind of pre-Darwinian orgy of the sharing of genes. And back then, and still with existing lineages, we see that the idea of working together was no big deal. Bacteria do that to this day. And some of our most pressing disease problems have to do with just how good they are at forming a we and then causing problems. However, one particular lineage over here, the eukaryotes, gave rise to a lot of interesting biology, including ourselves. So we get into something quite interesting over here, the evolution of individuality. So it's easy to be a we when you're just sharing genes and that's all that's going on. But when you start to evolve individuality, and individuality begat the brain, when you get into this area, now you've gone to me. You've gone from we to me. And me's have been very successful, but lots of problems also. But what I want to talk about are the later developments in animal evolution, the evolution of insect societies 150 million years ago, and of course lineages leading to our own. Now we all can agree that social evolution, the me to we part in animals, has given rise to some amazing traits that have changed the face of this planet for better or for worse: language, the ability to communicate, agriculture, and of course warfare. We also know that many of our afflictions and mental illness have to do with social interaction, social impairments. And so there's a devastating toll of mental illness, as you can see, and much of that is related to when the social brain goes awry. So it behooves us then to think about these issues of going from me to we. What can we learn about the social brain to be able to motivate ideas to eventually help treat these conditions. So I want to emphasize the evolutionary context to draw the problem out. There are advanced societies that have evolved in the animal lineages. Meanwhile, the bacteria and the archaea are doing just fine, thank you. They continued on their trajectory. But on the animal side, we have a very interesting situation. Only two groups of organisms have evolved complex societies: the primates, leading to humans over here, and the social insects, especially the ants, the bees, the termites, and the wasps. Social insects have evolved societies that, in terms of basic engineering principles, are every bit our equal in complexity. They have language, they have agriculture, they have warfare. So language, the dance language of the honeybee, which helps make it have an outsized impact on our environment, contributing to $30 billion a year in the US alone in pollination, all based on their social foraging abilities, aided by their language. And by the way, the last story I'll talk about today is about the honeybee dance language. Agriculture, the famous leafcutter ants studied by Edward Wilson, the founder of sociobiology. And these ants have a completely functioning agricultural system. They have fertilizer, pesticides, they harvest the fungus, they do everything that we know about modern agriculture, except little John Deere tractors have not yet been discovered in ant society. Otherwise, a full complement of agriculture 100 million years before our lineage gave rise to that. And then, of course, warfare. These are desert honeypot ants in southwestern Arizona, studied by Bert Holdobler, a close associate of E.O. Wilson. You can see that the ants are, in this inset here, standing, if you will, on their tippy toes, or tippy tarsi, if there are any fellow entomologists in the audience. And they're essentially checking each other out. The ants from the small colony, the small ants, excuse me, and big ants are checking each other out. The small ants go back to the colony and close all the doors to the entrance to the colony, prepare to get raided. The big ants go back to their colonies and prepare to mobilize a raid, where they go and attack colonies and take slaves, and then make slaves for the rest of the lives of those individuals. What appears to be happening there is that these ants can check each other out, and the small ones somehow know they're small, and then go back and communicate that it's time to batten down the hatches because there's going to be an attack. So all these forms are of activity are based on their sociality. Now, when you make these kind of comparisons, then it leads to this sort of question. And what I want to talk about today are approaches to frame this question in a scientifically more valid manner. Now, taking a crack at this question has been actually something of interest for a long time. People have been fascinated by social insects. Naturalists have looked at them and have wondered about this. However, the problem from the evolutionary perspective is as follows. So we have these two separate lineages, as I said, primates, humans, and the social insects. The last common ancestor lived some 600 million years ago. As far as we know, it was a marine flatworm with a very rudimentary nervous system, no brain to speak of, no social life to speak of. And so, by that, we can say, using the tools of traditional evolutionary biology, that these societies, although they resemble each other in some of their structural features, division of labor and segregated roles and so on and so forth, they had to, perforce, evolve independently. So if we want to ask the question of relevance to this symposium of what can we learn from the social insects that might be useful in thinking about human sociality, we have to ask, how is it possible to reconcile these independent evolutions? Well, what can we use as a history book to go back and assess this? Can we use the similarity of behavior? No. One person will say that aggression is similar in humans as it is to ants, and another person will say, no, it's not. And in fact, E.O. Wilson, who pioneered this area, used that level of comparison because that was all that was available in the mid-70s, and there was a split response. Some saw this as an intriguing comparison. Some saw this as a very dangerous comparison. Okay, let's drop down a level. Can we use brain? Unfortunately not. Even though the unit in the brain, the neuron, seems to be highly conserved across the invertebrate and vertebrate lineages, The functional organization at higher levels is not. That is, you can't point to a region of the vertebrate brain and look for its analog or homolog in the invertebrate brain very easily. Okay, so behavior, no. Brain, no. What about genes? Yes. We talked already, several of the talks have made use of the genome as a parts kit, as an instruction manual for interpreting biology, but the genome also has another very important role, and it does this very well. It's a history book. It's a history of life, and if you can read that book appropriately, you can learn many secrets about the history of life. And so it's possible to do this in a quantitative way. So the key concept, in case I forget to mention it later, is orthology. That is, you can look at a gene, the sequence of nucleotides that make up that gene, and you can ask if that sequence is similar enough to another sequence in another organism to be able to say, yep, I think that's the cousin of that gene, with statistical rigor. If you can do that, it opens up a whole world of comparative biology that is not available at the level of the brain or the level of behavior. So that's the framework that I will use to tell you about two stories today. First of all, nature builds diverse social brains with common genetic building blocks. So yes, the evolutions of societies have been separate based on the analysis that I just showed you, the phylogeny that I just showed you. But it appears to be the case that these separate evolutions can use the same genetic building blocks. And then armed with that knowledge, we can go a little deeper to look to see one particular aspect to be able to say that the social brain appears to be addicted to altruism. So let's go on. For the first study, this is a study that's been done at the Institute for Genomic Biology, a multidisciplinary thematic group. The entire institute is composed of such groups. So we have behavioral biologists, neurobiologists, computational biologists studying fish, stickleback fish, mice, and honeybees. And the idea is to look at a common property of sociality. So now we're not talking about all the fancy stuff in societies, but just a very common property of social behavior, which is social responsiveness, the ability to respond to socially salient stimuli. This is something that's much more widespread than society, so we started with this to see whether we could get some traction for the idea of looking for common building blocks. So all these species here that have rich social lives, all of them respond to stimuli that are either negative valence or positive valence. I'll be talking today about negative valence. That is a threat. They're able to identify a threat, whether it's conspecific, same species, or another species, label it that way, and then respond to it appropriately. So what we did is we presented a threat, we let the animal perceive it, we then let the biological processes take place at the neurobiological, at the genetic levels, and then studied those processes. And the idea was what we wanted to ask, using the tools of comparative genomics, whether we see a common basis for the response. That is, are there genes that are close enough that we can call them cousins, from bees to fish to mice, to say that, yes, the same genes are engaged. Some of you that come from the field of developmental biology may recognize the paradigm I'm talking about, because it started in developmental biology. It's the Toolkit paradigm. And this is a classic story from developmental biology, the Pack-6 story. So another feature of biology that is characterized by this convergent evolution is the eye, the vertebrate eye and the insect eye. Both form image-forming visions, and yet we know from phylogeny, like that phylogeny that I showed you before, they evolved separately. So any textbook, perhaps even the textbook still that Larry suggested that you all buy this morning, if you look there, it will say that the eye is a classic example of convergent evolution. And it is. However, no one told Pack-6, because you can take a Pack-6 gene from a mouse and put it into a transgenic fly, mutant fly that's blind, and that fly will now create an eye. And you can do the same for the fly gene, fly version of the Pack-6 gene, into a mutant, sightless, blind mouse. So the idea is Pack-6. When you need an eye, who are you going to call? You call Pack-6. I call it my Ghostbuster model. I'm really delighted that Ghostbusters 2 is coming out this summer, because the joke has gotten less and less understandable as I've gotten older and older. But now I'm going to be cool again once that movie comes out. So this is a classic story here. We're invoking the toolkit concept for the first time in social behavior. So just to restate what we did, we staged these experiments with social challenge, territory intrusion for sticklebacks, fish, the territory intrusion for mice, and then a colony intrusion, so a group of bees. You can't do anything with bees without being them in groups. They have to be in groups. Then we staged this, we validated that they were showing the normal response, and then we took parts of their brain, important parts of the brain that invertebrates have been known to be associated with social decision-making, and then a part of the brain in the honeybee, the mushroom bodies, that's involved in multimodal sensory integration, and so we reasoned probably was also involved in these kinds of things. And then we do, as you heard this morning, transcriptomics, global or genome-wide analysis of measuring the levels of gene expression. I want to just show you some summary results from these studies. So the take-home messages: are there genes that are common to bees, stickleback, and mice that change in their activity in response to a social challenge? The answer is yes. There are hundreds of genes that show this overlapping pattern. I want to just highlight some of the biological features that we can interpret, or biological processes that we can interpret by knowing the functions of many of the genes, and in most cases knowing those functions because we know the functions of the cousins of those genes in other organisms. So we see here a couple of key pathways. First of all, TF stands for transcription factors. We heard about some transcription factors already earlier today. These are genes that regulate the expression of other genes, sort of regulator genes. Many transcription factors are commonly engaged by a social challenge in these three evolutionarily distinct species, many of the same transcription factors. In addition, metabolism. Genes involved in metabolism are also involved. That is, they're differentially expressed, and they have a role to play in aggression. Many other pathways too, but I just want to highlight those. The slide, by the way, that you're looking at is a way of semantically classifying similar biological processes together to be able to get the overall picture. And you can see that there are clusters associated with metabolism. Each of these dots represents a set of genes that are commonly expressed in the three species, and that represent then a large swath of biology. And likewise, those of you interested in other parts of biology may cue in on those. But I want to highlight the notion of transcription factors and metabolism as being involved. So we have some evidence for toolkits for social behavior, and raising then some very interesting questions about how many of these genes are there? Are there specific themes in biology that are represented over and over again? I've highlighted two for you so far: transcription factors, that is regulating other genes, and metabolism. Are there different ones for different behaviors, or are there sort of Swiss Army knife toolkits that are repurposed for different behaviors, not only in evolution, but also within the organism and involved in multiple behaviors? Are there functions location-based? In neurobiology, it's all about location, location, location. So is it where they are expressed in the brain that influences what they do? These are questions that we are now mobilizing to pursue. But I want to take up one particular question, and that is, how good is the evidence of this toolkit idea based on an expression-based discovery or fishing expedition approach? So in Evo-Devo, in developmental biology, the concept of the toolkit has been instantiated by mutant approaches, knocking genes out and showing their function across species, like the example I showed you. That's sort of more hardcore molecular biology. This is a little softer. Changes in gene expression, really, you can use that evidence to infer toolkits. So I want to show you a couple of results that I think support the idea that one can use this omics approach to be able to infer toolkits for social behavior. And I want to mention results from 3 studies that I hope will give you that kind of information. First of all, a close relationship, between behavioral state and brain gene expression profile. I'll show you evidence for that. Secondly, I'll show you that it's possible to study the rewiring of gene networks and how that impacts behavioral plasticity. And the rewiring of gene networks, that's shorthand for saying changes in gene expression affect how genes are regulating each other. And then finally, an evolutionary insight based on sequencing the genomes of 10 different bee species, showing that the evolution of increased social complexity does involve changes in gene regulation. So the first one is summarized right here, and I chose this example because it also ties into one of the key biological processes that I identified for you before, and that's metabolism. This slide shows that there's a causal relationship between changing the expression of genes involved in brain metabolism, and for you cognoscenti, OxPhos, oxidative phosphorylation, is the particular type of metabolism that's changed when there is a response to an intruder. And we see evidence, causal evidence for a relationship between changes in brain metabolism and an increase in aggression in both honeybees and Drosophila. In honeybees, we did it pharmacologically. Our molecular tools for manipulating genome function in honeybee are progressing, but still way behind Drosophila and other classic model organisms. So honeybee's more of a model social organism. Drosophila is mostly home on Saturday night, not much of a Saturday social life. So in this experiment, we pharmacologically inhibited the oxidative phosphorylation pathway, both complex 1 inhibitor and complex 5, and we see an increase in aggression in response to doing that. In Drosophila, we did it genetically. We used genetic lines that downregulated a particular gene involved in OxPhos, also from complex 1, and you see an increase in the number of, in aggression, the number of lunges, relative to the 2 parental control lines that deal with some of the mechanics of Drosophila genetics, which I won't go over here. So to just close the loop, when you present a threat to a honeybee, stickleback fish, or a mouse, you get a decrease in oxidative phosphorylation. This is a process, a change in molecular change that goes on over minutes to even as long as several hours we tracked. You also see an immediate aggressive response, and we've also shown behaviorally that the animals, as you might expect, stay vigilant for a period of time. So the idea that the OxPhos decrease is then causing an increase in aggression makes sense when you think about the ecology of this particular social stimulus. That is, the presence of one threat often predicts the presence of another threat. And chronic stress syndrome is just the idea that the threat never goes away and has an extremely debilitating effect on biology. So, close relationship between changes in gene expression and behavior in a way that's relevant for what I showed you before. Number 2, transcriptional network rewiring. This is just a diagram to illustrate a little bit of the background of what I'm talking about. Genes are regulated by a suite of proteins, transcription factors, and cofactors that all pile up here in the part of the gene right before the part that actually makes the protein. They all interact. They will be stimulating the gene. In some cases, they'll be inhibiting the gene. It's a very complex process. That's what we refer to as transcriptional regulation. And what we have found is that one can put together gene expression analyses and build transcriptional regulatory networks to get an idea overall of how behavioral states are regulated by gene expression. And this was done with a large number of individuals, 853 individual brains, 48 behavioral states. But the point for today is to say we found some transcription factors that have global effects on gene expression. That is, they affect the gene expression, brain gene expression associated with different kinds of behavior, foraging, going out to collect food from flowers, aggression, what we've been talking about, maturation, the idea that bees go through a process where when they're young, they work in the hive, they do certain jobs. When they get older, they graduate and become foragers. That's a behavioral maturation process. We found some transcription factors that regulate all 3 of these neurogenomic states, and others that are more context-dependent, affecting only maturation and not aggression, for example. We picked 2 because they're in the same neighborhood of each other, that is, hormone signaling, and we know from Drosophila work that broad regulates FTZF1. These are the names of the 2. Broad affects gene expression in all contexts. FTZF1 is more specific, which leads to the prediction that FTZF1 regulates a subset of behaviors, and we wanted to test that because when you affect broad expression, you're also affecting FTZF1 expression because FTZF1 is underneath broad in a regulatory cascade. But we predicted based on this that there was modular effects such that we could affect broad and affect FTZF1 differently, as I'll show you. So what we did is we knocked down the expression in the brain using RNAi, RNA interference, of these two transcription factors. We predicted based on their role and their position in the network that both would affect behavioral maturation, and indeed both did. And let me emphasize that when you knock down broad, you are also knocking down the expression of FTZF1 because it's underneath it in a regulatory cascade. So the results that we got here made sense from that point of view. They also made sense from the point of view of the predictions from the network. So on the x-axis, age, on the y-axis, the number of bees that are growing up and switching from working in the hive to foraging, and then a suitable control, a molecular control for this knockdown experiment. You can see that knocking down the expression of these genes led to an earlier onset of foraging. We try to pick phenotypes, behavioral traits, that show what's called a gain of function, because knocking down the expression of a gene with RNAi in the whole brain is a very crude method. In Drosophila and mouse, it's possible to do that with much better control. So we have to guard against off-target effects. So our rationale is we'll look for a behavior that's showing an enhancement in some way. So the idea is, okay, bees are starting to forage at a younger age. They're doing the riskiest, the most demanding, the most cognitively difficult job. They're going out, flying around 15, 20 minutes, making hundreds of stops at flowers. When they finish, they make, pardon the pun, a beeline back to the hive. They know where they're going. So it's hard to imagine that causing that earlier in life is an off-target effect. So we see this. By the way, we quantify this using RFID tags that are affixed to bees, and we have an entrance to be able to read the bees, so that we basically, a few years ago, traded in sunscreen for this system here to be able to get all the records of the bees. So knocking down broad and FTZF1 causes an early onset of maturation. It affects both. But the same approach only affected aggression for the broad knockdown and not for the FTZF1 knockdown. This is predicted by the modular effects in the network, not predicted by the fact that when we knock down broad, we are knocking down FTZF1. So here we see the results of a dish assay for aggression, and it's really great to be able to talk about this assay here at the University of Colorado because it was developed by Professor Michael Breed some 30 years ago and has been used in many labs and has been a great workhorse for many, many different scientists. So thank you, Mike. So here we see the aggression index on the y-axis, and you can see when you knock down broad, you get a significant decrease in aggression, whereas when you knock down FTZF1, no effect. So without belaboring that, I will say that we see similar effects on brain gene expression as well, supporting the idea that this rewiring of transcriptional regulatory networks has phenotypic consequence in the context of social behavior. So then the last example here before switching to the second and final story is the evolutionary story here, which is that the evolution of increased social complexity involves changes in transcriptional regulation. To do this, in cooperation with BGI in China, we sequenced the genomes of 10 different species of bees that range in social complexity from solitary to a little bit social, a little bit more, all the way up to the pinnacle of sociality, the honeybee. And what you see here is a complex slide, so let me walk it through you. Walk the slide. You know what I mean. We are looking at the number of significant correlations between the number of transcription factor binding sites in the promoter region of genes, the same genes across all 10 species. These transcription factor binding sites, if you go back to that cartoon that I showed you, are the places where the transcription factors dock and then regulate the expression of the gene. They have a particular signature. You can scan for them using computer algorithms and identify many of them with reasonable confidence. So we used that scan across the 10 species. We used an algorithm developed by Professor Saurabh Sinha, who was in part of our team at the IGB, and counted the number of transcription factor binding sites for specific transcription factors in each promoter. Scale that along the axis of increased sociality. And you can see there's a predominance positive correlation. That is, as you increase sociality, you increase the number of transcription factor binding sites for specific transcription factors. Not all, some showed a negative correlation, but the overwhelming majority showed a positive correlation. Functional significance of this is not known, but we might hand wave and say that if you just think in engineering terms, the more contact points, think of a rheostat, the more contact points you have to regulate something, the better you're able to regulate it. And while many can say very reasonably that by the time a stimulus from the environment is transduced and goes all through the various biologies that get you into the nucleus of a cell, it really shouldn't matter whether it's heat or light or a touch or anything else. As a social behavior snob, I would say it probably still does matter. Coordinating behavior among individuals is complicated, and it's intriguing to think that there's a molecular correlate or base to that into that increased sophistication of regulation. So those are the three examples to help make the point that using gene expression approaches are valid for studying toolkits, for looking for toolkits, because basically of the close relationship between changes in gene expression and changes in behavior. Okay, so then the second and final point is based on the idea then that one can think about common molecular building blocks that scale, that go across. Can we go deeper than that and look at biological processes that are more integrative than, say, metabolism? Can we go up a level of biological organization? So I don't think we have any problem thinking about this, this concept right here. What about this one? How far can we go with this? When we think about some of these social behavior traits, can we think about the fact that since they are motivated by altruism, that the altruism actually is instantiated in the brain with processes that we recognize as being as the kinds of changes in the brain that go on during addiction. So let me motivate this a little bit further. When a solitary insect finds good food, she does what you might expect. She'll eat more. But when a honeybee finds good food, she does not eat more, she dances more. The dance is a symbolic communication. She goes back to the hive, she tells others about the food, and she motivates and mobilizes her sisters to go out and collect the food. So that's an interesting me-to-we behavioral switch right there. This is the famous honeybee dance language discovered by Carl von Frisch, for which he won a Nobel Prize in 1973. This dance is able to communicate distance, direction, and value for the food. So bees go out, they collect food, they come back. If it's of a particular quality, they come back and then communicate information that's encoded distance, direction, and value of the food. Experiments with robots have shown that honeybee communication is about 50% effective. As someone who's married almost 35 years, 50% is very good. I'll take it. So it's a very effective communication system. And I want to show you how we've been using the dance language to be able to explore this idea of addiction. Is altruism, can it be seen in some ways as in an addictive context? So the first experiment is to, again, do another transcriptomic experiment. We measured gene expression, and we looked at bees that were given food in the laboratory, just one-on-one, just given a little bit of food, and then after a certain amount of time, measured changes in gene expression in, again, that region of the brain, the mushroom body. Other bees, we trained to a nice feeding station, allowed them to collect food, and made sure that they liked it enough, and they showed us they liked it by dancing. And then we collected them after a suitable amount of time after dancing. Again, this is sort of a very schematic summary. We found over 1,000 genes differentially expressed when bees were given, as in individuals, a little bit to eat. We found several hundred genes that were engaged, differentially expressed after a bee danced. And we see significant overlap. These were the large circles, so using network-type analysis that involve clustering genes based on similar patterns of expression. Anyone interested in that, I'll be happy to talk about it later. We see modules of genes that are showing common patterns of expression, say food 1 and food 2. We did the same thing for the dance genes, dance-responsive genes, dance 1, dance 2, dance 3, dance 4, and the presence of the red circles inside the blue circles mean there was a significant overlap of the genes engaged by dance as those that were engaged when the bees were just given the food. Over here, we see a neuroanatomical analysis using an early immediate gene, c-Jun. We looked for places in the bee brain. This is a schematic, a coronal section of the bee brain. Looked for places in the brain that were lighting up in response to these particular stimuli. The blue are parts of the brain that lit up either when the bee was given food in the laboratory as a solitary individual or after the bee started to dance. So neuroanatomical overlap and then molecular overlap. By the way, we used a tunnel to be able to do these experiments, which is going to be particularly important in the final experiment that I'm going to tell you about. This tunnel is very important because what we were able to do is trick bees to think that they were flying further than they really had. That's important because the dance language is calibrated based on distance. When bees fly a distance of 100 meters or more, they embed in that dance exquisite directional information. When the distance is very short, their communication is much more rudimentary. It's along the lines of, there's food out there, it's close by, go find it. And much less precision of that. We wanted to use their best dances because, as I'm about to show you, we manipulated using neuroactive substances their brain, and we wanted to be able to, again, look for precision of effects. So if we see a change in the likelihood of dance, we want to be able to rule out that it was just a motor program change. Because where I'm going with this is biogenic amines, cousins to dopamine, and as many of you know, dopamine is both pleasure juice and Parkinson's, motor. So these brain chemicals have multiple functions. We wanted to be able to have a phenotype that we could separate them. So we took advantage of this really great discovery of Manyan Srinivasan, which is that the way bees measure distance is by optic flow. So the more information that flows past their retina, then the further distance they think they've gone. So you can fly bees through a tunnel, and if it's a dense checkerboard pattern, when they get to the end of the tunnel and they come and they collect the food and they go back home, they go, wow, that was a long distance. They will dance as if it's a long distance. If you change the walls of the tunnel and make it a sparser pattern, less visual information, same bee, same colony, same food, they come back and they go, nothing, piece of cake. So we tricked the bees because we used this enclosed flight room here, outdoor flight chamber, to have natural exposure, but also to be able to control what food they ate because we were spiking it with chemicals. I'm not going to tell you what chemical I'll fix until the next slide. So first chemical that we used was octopamine. It's a cousin to dopamine. And octopamine makes solitary insects eat more. So we figured if there was a chemical that could instantiate this me to we switch, it would be octopamine. And in fact, it was. So here are the controls. The octopamine was administered in a particular solvent. We gave bees lousy food that would give them a very low likelihood of responding by dances. So less than 10% of the bees found favor in the food to dance. That was to avoid a ceiling effect, just experimental stuff. So then, giving them the octopamine, we could triple the number of bees that were dancing. Now, at this point, to invoke the idea of the reward system is still a little bit of a stretch because octopamine and the concept of a reward system in insects is not as well developed as it is in vertebrates. So we decided to take a little jump and go to cocaine. And we find that cocaine also makes bees dance more. It gives the same effect as it does octopamine. For you pharmacologists out there, you will recognize this as a trivial experiment because coca exists in nature as a natural insecticide to engage and agonize, the insect's octopaminergic system. It's an octopamine agonist, and herbivores that munch on plants that contain the substance have a severe problem because their system is overstimulated and they die. So we knew that, so it wasn't such a big leap. But still, no one can argue that cocaine, the bona fides of cocaine in reward concepts are pretty solid, for better and for worse. And so we thought that we would do this experiment because you never know, stranger things have happened. So we expected to see it would have the effect, and it does. When we block the octopamine receptor pharmacologically, we block the cocaine effect, and so we feel fairly confident to the limits that neuropharmacology allows us to say this, that we have an effect that's fairly specific and that we have engaged the system to create bees that are more likely to dance by increasing their ability to value this particular food. And so this engages them, and then you get this altruistic behavior. So these me-to-we mechanisms, just wrapping up, they may provide insights into addiction. They can also explain the delicate balance between cooperation and competition. And I think an insight that's worth looking into further, both in our societies as well as in insect societies, the idea that there's a very thin edge, thin razor there, separating selfishness from altruism, and that may be actually at the biological level as well. So just in general, let me wrap up by saying bees are not little people. Don't come away with that conclusion. But nature has used common genetic building blocks to repeatedly evolve social brains. And then one early insight that's based on that idea is that the social brain is addicted to altruism. Let me acknowledge the individuals whose experiments that I spoke about, as well as collaborators and funding agencies. The Toolkit idea, by the way, Tom, was funded by the Simons Foundation because NIH thought it was a fishing expedition. Thank you very much.
Okay, we have questions way up there. So you need a...
Thanks so much. Jean, very much enjoyed your presentation. Can you translate the complex 1, complex 5 inhibitor that you used and the link into oxidative phosphorylation into rodent mutants in complex 1 and their behavioral changes compared to wild-type rodent lines? There are quite a few available, and there may be links there. Can you expound on that?
Yeah, not off the top of my head. I'm familiar with that work, and I believe at least several of the mutants are in complex 1, but you sound like you might know better. Do you?
There are several mutants in complex 1, and I'm just paining my brain to remember whether comparable mutations in humans, like in Liebers, Elhon, and others, they are associated, I think, with reduced social interaction, also lack of empathy, and it's quite a stretch, so it's a difficult topic to kind of like broadly philosophize about, but the mouse might be an inroad.
Yes, yeah, no, that's a great point. Love to talk with you about that more.
So I've read lots of E.O. Wilson, and I've heard your talk, and I'm confused about one thing, and well, more than one, but at least one. So this definition of social, the we thing, those of us that have pets might have included dogs as social animals because they hump each other or whatever. So I'm confused about what E.O. Wilson and you talk about this convergent evolution. I need more clarity that would help me. Can you do that?
Sure, yeah, that's a great point. And these represent extreme societies. And in fact, there is a broad continuum, especially in the social mammals. There are societies that have some elements, but there's a key difference. And in fact, there's a key difference that separates the social insects even from human society, and that is a reproductive division of labor. So there is, as you can imagine in biology, a taxonomy for this, and there are 3 criteria that define the most extreme form of sociality called eusociality, and that is cooperative brood care. So you could say many species have that, including ourselves. Overlapping generation of adults, also. The third one is reproductive division of labor, where some individuals reproduce and some never do. Some human biologists or human scholars will say that that's sort of the case in humans, but generally speaking, not really. And so that's even something that distinguishes the social insects. Otherwise, there's just a broad continuum.
Thank you, Gene. It's more of a layman's question, not getting into the molecular genetics of the process, but we kind of understand there's a problem with the colony collapse with the bees, and it's kind of an ongoing issue. And I was just wondering, with the antagonist and the research you've done on that, as far as the genome is considered, with the evolution of the bee species and the socialization, when they're going out and harvesting their pollen and their foodstuffs, is it possible to somehow put an antagonist into that genome, into the food crop, like corn or something like that, so that it's not contributing to the colony collapse? Maybe there could be like a warning to the bees to maybe stay away from that particular food source?
Yeah, so the question had to do with colony collapse disorder, which is a malady affecting bees worldwide for the last 10 years. And the general gist of the question is, can genomics be used to help address this problem? And the answer is yes, it has been. It has helped identify some of the pathogens that are causing this. Our basic understanding now of colony collapse disorder is that it's multifactorial. It involves parasites, pathogens, pesticides, and poor nutrition, acting in as yet unexplained and undiscovered synergistic ways. Sometimes one is a stronger factor than the other. Any of you ever tried to do combinatorial experiments know how difficult it is to tease these pieces out. And genomics has been used in many different ways to help move this research forward.
So one last question. How about Frank here?
Hi, thanks very much. Almost an extension of the last question. Very interesting. I think what you're saying, being a layperson, is that there's really a delicate balance here in terms of the molecular driver behind, in a sense, social behavior. That social behavior is critical to survival of a particular species or group, whether it be bees or human beings. And then the question is this, it goes back to Gene Roddenberry, for those of you who remember Star Trek, and they arrive at a planet where there was pollution and there were only 4 deranged people left because they had polluted the planet and people began to change. They became very violent in this case. What have you begun to look at the issue of environmental stress? For example, we have this thing called global warming going on. And doesn't that raise that delicate balance, that you're observing, doesn't that raise a great deal of questions and concerns going forward, not just for bees, but for ourselves?
Yeah, it's a very interesting question. Just 2 days ago, our group put in a large proposal to take the Toolkit concept further and look at how does early stress affect the robustness of these collections of genes that are responding across the species. Does it tighten them up? Do some fall out? Are some categories differentially expressed? So we know, as Phyllis mentioned in my introduction, that environmental conditions affect brain gene expression in profound ways, and really changing the way we understand nature and nurture as a result of that. And so various forms of stressors will be having effects on behavior through their effects on genomic activity in the brain. It's harder to study, but there's, based on inferences from the animal models, there's no reason not to expect that.